Hello friends, I am Dr. Amit Gangwal, Associate Professor at Shri Vile Parle Kelwani Mandal's Institute of Pharmacy, Dhule. In this video, I will talk about the first chapter of B.Pharm, 5th semester subject Pharmacognosy and Phytochemistry 2. And the topic is unit first, study of utilization of radioactive isotopes in the investigation of biogenetic studies. So on this slide, I will spend 5 minutes. Biosynthetic pathways or biogenesis means the entire marathon or the entire journey of formation of various primary and secondary metabolites in the plant. Plant में primary और secondary metabolites कैसे बन रहे हैं? How the study, how this journey is traced? It is called the tracer technique, because you are tracing or you are tracking or you are following the pathway or journey of formation of C from B or A to C. How different types of primary and secondary metabolites are formed in the plant from various precursors. It is subject matter of this particular video. So there are few definitions. Let me clarify those terminology first. Biosynthetic pathway, the chemical series of chemical events or series of chemical reactions in the living system is called biogenesis or biosynthesis, or different routes or pathways are called biosynthetic pathways. We all know plants give two types of metabolites, primary and secondary. Primary means they are of prime importance, they are very important, so they are called primary, and examples are carbohydrates, lipids, and amino acids. So carbohydrates consist of monosaccharides, disaccharides, and different types of higher jumbo molecule like polysaccharides, for example starch, pectin, cellulose, and others, or glycogen for that matter. In case of proteins, they are consist of amino acids. We all know two types are there: essential and non-essential amino acids. Essential means they are to be given to the human being from outside, because humans can't design, I mean they cannot make all the amino acids in the body, so they're called essential. Third is fatty acids or fats or lipids. So these are three primary metabolites which are formed first in the plant, so they are being called primary metabolites, and these primary metabolites synthesize secondary metabolites. So what is the role of primary metabolites in the plant? Primary metabolites are only responsible or essential for normal growth, development, and maintenance of the plant, and secondary metabolites are designed in the plant by primary metabolites only. So what is the role of secondary metabolites in the plant? Secondary metabolites as such have no major or any breakthrough role in the plants. They are basically used by human beings for various purposes. One of the most important reason is in the treatment of various disorder. So example of secondary metabolites are alkaloids, tannins, terpenoids, glycosides, resins, tannins. Few people called polyphenolics. So phenolics or polyphenolics, they are very broad category. They include lignans, tannins, and flavonoids also. Then in flavonoids, we have different types of flavonoids like flavin, flavanol, flavanone. Depending on the moiety, there are different types of flavonoids. Clear? These are the example of secondary metabolites. Now, how to study the route of biosynthesis? The precursor or the starting material is attached with a radioactive material. For example, you have to study synthesis of atropine. So you all know ornithine is the amino acid which is responsible for biosynthesis of tropane. Tropane is the chemical structure, chemical moiety present in the atropine. सो ट्रोपेन के बनने में किसका रोल होता है? ऑर्निथिन का. तो यू विल अटैच वन radioactive isotope in the ornithine molecule, and the moment you attach a radio labeled component or the moment you replace one of the carbon or nitrogen of ornithine with isotope of carbon or nitrogen, it will be called radio-labeled precursor or radio-labeled ornithine. So the first step is decide the precursor. It is ornithine. Then attach radio-labeled component or the replace one of the carbon or nitrogen or other atom with the radioactive isotope of carbon or nitrogen or other atom of the interest. The moment you do this, it will be called radio-labeled precursor or radio-labeled ornithine. Now second stage is to incorporate this radio-labeled ornithine into the plant, that is feeding, to feed, to incorporate in the plant. And after the incorporation, when the sufficient quantity of the atropine or tropane has been made in the plant, you will extract, you will isolate the material, you will check its radioactivity. If it is giving radioactivity, then you can have some idea कि tropane or atropine has been derived in the plant from ornithine only. So what we have seen, meaning of tracer technique, feeding, incorporation, meaning of biogenesis or biosynthetic pathways, and definition of primary and secondary metabolites. We will go ahead now. So content in this video has been taken from books like AK Set, famous book of cognosy worldwide, Treason Evans, and small books highly customized as per the syllabus of PCI by Nirali Prakashan. These are the books by Nirali Prakashan, and one is by Thakur Publication. So what are various pathways for formation of primary and secondary metabolite? So we all know, in presence of sunlight and carbon dioxide, by taking water from the soil or the ground, food is prepared in the plant through photosynthesis. So photosynthesis is the process in which carbon dioxide, water in presence of sunlight react and they give the first jumbo molecule, that is glucose, along with some other molecule. So this glucose become part of, this is the glucose, I mean the carbohydrate obtained from the photosynthesis. It will become part of glycolysis and pentose phosphate pathway. Two routes are there for the metabolism of glucose in the plant. One is glycolysis, another one is pentose phosphate pathway, PPP or HMP shunt pathway. So in glycolysis there is formation of phosphoenol pyruvate, and pentose phosphate pathway will give erythrose 4-phosphate. These two are the starting material for the shikimic acid pathway. Shikimic acid pathway will give aromatic amino acids, that is tryptophan, tyramine, tyrosine, and phenylalanine. These are three aromatic components, and these three aromatic amino acids are utilized in the plant for the formation of alkaloids. Right? Now, glycolysis will give pyruvic acid, which will convert into acetyl-CoA. Acetyl-CoA will take two pathways. One is acetate malonate pathway, another one is acetate mevalonate pathway. Acetate mevalonate pathway will give terpenoids, isoprenoids, and steroids, and all type of components having jumbo structures like pentacyclic triterpenoid or steroids like cholesterol, and all this jumbo molecule resembling to steroids. And the precursor for steroid synthesis is squalene. Now acetate malonate pathway will give you fatty acids, polyketides, and aromatic compounds. Right? This is the summary of entire biogenesis or biosynthetic pathways in the plant. This particular cycle or the structure is the summary of how different types of primary and secondary metabolites are formed in the plant. In coming session, I will talk more about this. Summary again in different manner. So CO2 H2O will give the glucose. This is through photosynthesis, and this is the primary carbon metabolism. So this will give you erythrose 4-phosphate from phosphate pentose pathway. Phosphoenol pyruvate will be obtained from glycolysis. So there are two cycles for the metabolism of the glucose in the plant. One is glycolysis, which will give phosphoenol pyruvate, pyruvic acid, and acetyl CoA. Erythrose 4-phosphate will be obtained from phosphate pentose pathway. These two are the starting building block for the starting of the shikimic acid pathway or shikimate pathway. Shikimic acid pathway is well known for the designing or the biosynthesis of aromatic amino acids in the plant. And these three aromatic amino acids are tryptophan, tyrosine, and phenylalanine. These three are utilized for the biosynthesis of various types of alkaloids. We all know alkaloids are secondary metabolites, right? Now, as we have discussed, glycolysis cycle will give pyruvic acid and it will give acetyl CoA. Acetyl CoA will be utilized in two pathways. Just we have discussed acetate malonate pathway and acetate malonate pathway. Malonate means it will give you polyketides and fatty acids. Malonic means it will give you steroids, terpenoids, cholesterol, isoprenoids, and other type of hormones. So already we discussed the difference between primary and secondary metabolites. Primary means these are the biomolecules required for basic metabolic processes. Primary are generally present in all type of plant irrespective of the family, genus, species or type of plant. On the other hand, secondary metabolites are highly specific to particular plant or some particular species or genera. For example, if you talk about opium, it is not present in all the plant. It is mainly present in Papaver somniferum. Similarly, if you talk about quinine, it is mainly given by the god only in Cinchona species, Cinchona genus and different species of Cinchona. So secondary metabolites have limited role in the plant. For example, they are source of nitrogen reservoir in case of proteins are to be designed. They are the waste product. They are the toxic material. So different types of cattle which graze on the plants are away from those type of plants. Already we have discussed this. Then this is the glycolysis. As I told you, glucose will come from photosynthesis. It will become part of glycolysis cycle. It will give us phosphoenol pyruvate. It will give pyruvate and it will further become acetyl CoA. This will be utilized for the shikimic acid pathway. So what is important in all these pathways, you have to remember name of the precursor, name of the intermediate, name of the final product, name of the enzyme also, and type of energy release. So in this particular case, the cycle name is glycolysis. What is the precursor or the starting material? Glucose. And these all are the intermediates, and this is the end product, right? And these are the enzymes responsible for each and every step. So we all know there is no meaning of biosynthesis. They cannot be designed in the plant if there is no enzyme. So in case of synthesis in the laboratory, we have different type of catalysts. Similarly, in plant, we don't have catalyst. We have the God-given enzymes that you have to remember in all this cycle, right? And these are the energy, that is bioenergetics. कितना molecule बन रहा है, कितना ATP निकल रहा है. This is the bioenergetics, which is important from examination point of view. Now, this is another pathway for metabolism of glucose. As I told you, the first one was glycolysis, which will give you pyruvic acid and phosphoenol pyruvate. In case of HMP shunt pathway, which is also called phosphate pentose pathway (PPP), it will give you erythrose 4-phosphate, and this will be utilized for the production of shikimic acid molecules. So this is Shikimic acid cycle, it is very easy now. This is phosphoenol pyruvic acid coming from glycolysis. Erythroid 4-phosphate coming from HMP pathway. This will give you 2-keto 3-deoxy 7-phospho-D-glucoheptonic acid. Likewise, it will give you different types of chemical. This is shikimic acid, the main name of the cycle. It will give you crocimic acid, it will give prefenic acid, and here there is bifurcation. This is the phenyl pyruvic acid and this is para hydroxyphenyl pyruvic acid. One will give you phenylalanine, another one will give you tyrosine, and third one is tryptophan. So this tryptophan, this tyrosine, this phenylalanine, these are the important 3 aromatic amino acids designed in the plant only through shikimic acid pathway initially. The same reaction in different manner. Okay, so this is very beautiful picture. I mean, shikimic acid will give chlorogenic acid, then crocimic acid, phenylalanine, tyrosine, and now tyrosine is the aromatic amino acid obtained from the shikimic acid pathway. But in how many ways? What are the chemicals which are designed by, or what are the chemicals in the plant, or what are the secondary metabolites which are biosynthesized by the tyrosine? These are the names. Different types of alkaloids, then betalains and tyramine and hordenine, this one like this. Similarly, tryptophan will be utilized for the biosynthesis of indole alkaloids. Indole alkaloid means Rauwolfia serpentina, reserpine, then vinca alkaloid, that is vincristine, vinblastine. So they are the example of indole alkaloids. Indole is a heterocyclic ring which is done in the plant by tryptophan. Tryptophan is coming from shikimic acid cycle. So we have seen HMP pathway, we have seen glycolysis, we have seen shikimic acid cycle, we have seen starting cycle, that is photosynthesis. Now we will talk about acetate pathway. Glycolysis will give you pyruvate which will become acetyl CoA. Acetyl CoA will become part of acetate mevalonate pathway and acetate malonate pathway. There are two options, and this will give malonic acid, it will give isoprenoids and squalene. Squalene is the first jumbo intermediate or the precursor for the formation of steroids and I mean saponins and other type of structures resembling steroids and saponins, and it will also utilize for the biosynthesis of higher terpenoids like rubber and carotenoids and others. On the other hand, if we talk about acetate malonate pathway, it will give you malonyl CoA and it will become long chain fatty acids and lipids and fats and waxes. This is how this is utilized in the plant. Already we have discussed. Purposely, I have taken various infographics and charts from various sources from the internet by using Google as search engine. So this is very important. This is the cycle responsible for formation of or biosynthesis of isoprenoids or terpenoids in the plant. The starting material is acetyl CoA, which is coming from pyruvic acid, which in turn coming from glycolysis end product. So acetyl CoA will react with the acetoacetyl CoA. It will become hydroxymethyl glutamate. It will give you mevalonate, and it will finally give you isopentenyl pyrophosphate and its isomer dimethyl allyl pyrophosphate. These two will unite and they will give you the geranyl pyrophosphate. One more molecule of IPP, that is isopentenyl pyrophosphate, will be mixed with GPP, that is geranyl pyrophosphate, and it will give you farcinyl pyrophosphate. Likewise, cycle will go on and you will get monoterpene, diterpene, sesquiterpene, tetraterpene and terpenoids of higher category. This one. So this is the starting material, isopentenyl pyrophosphate. It will react with its isomer dimethyl allyl pyrophosphate. They will unite together. They will give the first component, that is geranyl pyrophosphate, C10. It is monoterpenoids. Here it is mono means one, but in case of terpenoids it is 2. So 2 units of C5H8 will be called monoterpenes. C5H8 is isoprene or terpene. the basic or simplest unit of isoprene or terpenoids. Example of monoterpenoids are menthol, limonene, geraniol, linalool, and citral. We all know that sesquiterpenes and monoterpenes are the main chemical components present in the volatile oils. So IPP means isopentenyl pyrophosphate. Basically, it is C5H8, single isoprene unit. So when this is 2, now in GPP, that is in C10, you are adding one more C5, that is IPP. It will give you C15, that is sesquiterpene, and it is called farnesyl pyrophosphate. Likewise, you will go on adding IPP and you will get higher type of chemicals, that is called triterpenoids, polyterpenes, rubber, lycopene, beta carotene. They are the examples of C40, that is tetraterpenoids. Tetra means 4, but here it will be 8. So C5H8 8 times will give you C40 structure, that is tetraterpenoids, and the examples are lycopene, beta carotene, and others. Same cycle in different manner. Same. Now this is the uh, biosynthesis of protein. So different types of amino acids are biosynthesized from the plant. Very clearly you can see from this particular cycle. They are self-explanatory. Total 6 families are there. Amino acids of the serine family, amino acids of tryptophan category, glutamate category, and aspartate category. And this is the basic metabolic pathway. Already we have discussed. This is TCA cycle. So different portions leave the cycle and they become part of biosynthesis of amino acids. So already we have seen. In this way, we finish the biosynthetic pathways, primary and secondary metabolites, and their studies how they are designed in the plant through tracer technique. Now we will talk about methods in tracer technique. There are 3 methods: precursor product analysis, sequential analysis, and competitive feeding. Precursor product analysis, sequential analysis, and competitive feeding. All the names indicate exactly what the process is. For example, in precursor product analysis, we insert or incorporate a chemical compound which is presumed precursor. It means the compound which is supposed to convert into the product or the secondary metabolite. Based on the structure of final secondary metabolite of the interest, we decide which type of precursor is most suitable for the biosynthesis, right? So we study precursor and product and the intermediate. So for example, in case of enabasine, which is a secondary metabolite obtained from the Nicotiana glauca plant, we have to insert lysine. So suppose lysine, what happens in this precursor product analysis method, you have to insert compound A and it will give you C. So A is the precursor or the reactant or the starting material, and C will be the product or the secondary metabolite. So there are 2 chances: either you will get C directly from the A, or A will become the part of general metabolic pathways and it may produce B, D, E, and other so many compounds, and from those so many compounds or the intermediates you may get C. So we don't know exactly A is giving C or different metabolites obtained from A will give C. So here there is role of multiple labeled compounds. So already we have discussed earlier, there are need of labeled compound, means the precursor will be attached with the radiolabeled component. I tell you. So here in this case you are saying this is the structure of lysine. So it is the presumed precursor or the tentative or probable precursor for the formation of this ring of enabasine. So this is the precursor or the starting material, and this piperidine is the end product or the secondary metabolite. So we have to study what type of nitrogen is coming from this ring to this ring. So this is our lysine. Lysine is the presumed precursor. So this particular carbon will be replaced by the isotope of carbon, that is by radiolabeled carbon. So the moment we replace this normal carbon with the radioactive carbon, it will be called radiolabeled lysine, because lysine is having a carbon which is not the normal carbon, but it is the radiolabeled carbon. So this is the lysine in which this particular carbon has been replaced by the isotope of carbon, that is 14C. And here this nitrogen is not the normal nitrogen. This is the isotope of nitrogen. So now in this product, that is enabasine, this piperidine ring of the enabasine, what it indicates? It indicates that the same nitrogen, sorry, the same carbon which was replaced by 14C has been found in the product also. This is the symbol of radioactive carbon. Similarly, this nitrogen is also the same nitrogen which is coming over here. It means the this particular carbon and this nitrogen have, I mean, they are they came from the lysine. It means the C and nitrogen both were replaced by isotope of the carbon and nitrogen respectively. Both these atoms have been found in the piperidine ring of enabasine. It means the nitrogen and this carbon have came from the lysine. In another study, you see this one. Now in this study, this carbon has been replaced by radioactive carbon, that is isotope of carbon. Similarly, this nitrogen attached with this particular carbon has also been replaced by isotope of nitrogen. Now when we insert this lysine, or when we feed this lysine, or when we incorporate this lysine into the N-glauca plant, there will be formation of the enabasine. So our point is, in this ring of enabasine, that is the piperidine part of enabasine molecule, this carbon is again coming from this carbon. As such, means this carbon has been coming from this carbon, but this nitrogen has not been utilized for this nitrogen. Had this nitrogen of piperidine ring come from this ring, it means it would have been isotope. But very clearly we can see the nitrogen of piperidine ring of enabasine is not isotope. It is the normal nitrogen. It means this labeled nitrogen has not been utilized in the biosynthesis of enabasine. So in this way, by multiple labeled compounds, we can come to conclusion what type of atoms of particular molecule is being utilized. One thing is clear, that lysine is the precursor for the biosynthesis of piperidine ring of the enabasine. But here, what is the essence, what is the bottom line, what is the take home message? In this lysine ring there are two nitrogen, but which nitrogen is being utilized for the nitrogen of piperidine? So we come to conclusion that the nitrogen which is at this position, this one, is utilized for the formation of piperidine ring of the enabasine. This is how things are confirmed in case of precursor product analysis. Now we will go to second methods, that is competitive feeding. What happens? As I told you earlier, A is the reactant and C is the product. So A will give B and A can also give B dash. So exactly they are the intermediates B and B dash, A to C, but the route is not directly A to C. Route is A to B or A to B dash, and either B will make C or B dash will become C. But exactly B or B dash, which one are being utilized for the formation of C? In this case, in one plant, we will give B and the plant will produce C. In another plant we will give B dash and it will give C. So if in the C we get B dash, if your B dash is found in the C, I mean if B dash is labeled and it is giving C, and if C is also giving radioactivity, it means B dash has been utilized in the formation of C. But if C is not radioactive, it means B dash has not been utilized by the C in the plant. In this way, we can jump to conclusion that the intermediate which is having radioactivity, if the radioactivity is found in the C also, we can conclude that the C is the product which has the B dash in it, because C dash is also showing radioactivity. So the example is tyrosine gives 3,4-dihydroxyphenyl pyruvic acid. So there were two assumptions initially, that tyrosine can directly give 3,4-dihydroxyphenyl pyruvic acid, or tyrosine will become dopa and dopa will become 3,4-dihydroxyphenyl pyruvic acid. But by the study, as I told you, that is in competitive feeding, it has been found that tyrosine directly give the 3,4-dihydroxyphenyl pyruvic acid. But yes, tyrosine also gives dopa, but dopa will never become part of 3,4-dihydroxyphenyl pyruvic acid. Means it means tyrosine is the direct precursor for the biosynthesis of 3,4-dihydroxyphenyl pyruvic acid. There is no role of dopa in the biogenesis of 3,4-dihydroxyphenyl pyruvic acid. And the last is sequential analysis method. As the name indicate, if we have to study what type of all the possible chemicals are biosynthesized in sequential manner from the reactant or the precursor. For example, in the plant of Mentha piperita, piperitone will give this piperitone will give menthone, this will give menthol. So if one of the carbon atom, for example, is labeled by is replaced by radiolabeled carbon, then if again it is giving menthone, and menthone is also showing radioactivity, means menthone has the carbon which is radioactive or isotope of carbon, and it has come from the piperitone. Similarly, menthol is also giving radioactivity. It means menthol is possible because of menthone, which in turn is possible because of piperitone. So when we have to study the continuous route of different type of intermediates and the products from one single chemical moiety, we have to go for sequential analysis method. In this method, what we do, the principle of this method is to grow plant or the species in atmosphere of 14 CO2. Means we all know plants require carbon dioxide. So when you are growing a plant or plant in the atmosphere having 14 CO2, means carbon dioxide, in which C has been replaced by isotope of carbon which is radiolabeled, which is radioactive isotope of carbon, then all the plant which require all the molecule which require carbon will be showing radioactivity, because they have taken the C from this 14 CO2. So what they have mentioned, best way is to grow plant in atmosphere of 14 CO2 and then analyze the plant metabolites. Isolate all the plant metabolites, isolate menthone and check the radioactivity, isolate menthol and check the radioactivity. It means they are becoming possible in a sequential manner, and that's why the process is called sequential analysis method. Example is, I told you, keep Mentha piperita plant in atmosphere of 14 CO2 for 5 minutes, and it will provide the evidence of probable biosynthesis sequences. So in this way, we have finished biosynthetic pathways, what is the definition, what is the meaning of the word, what is isotope, and what is the need for biosynthetic pathways, different primary and secondary metabolite formation cycles, and one more thing we have discussed, the role of isotopes and radio-labeled material in the study of biosynthetic pathways or tracer techniques. Thank you very much.